Welcome back to Hunting Truth. In a world awash with information, some stories twist and turn, morphing from scientific fact into widespread fear. Today, we re-unraveling one of those stories, a story about a little-known acronym that has sparked outrage, boycotts, and fervent online debate, HEK 293. You might have seen the headlines, heard the whispers online, or perhaps even seen videos claiming that HEK 293 is lurking in our vaccines, our medicines, and even, most controversially, our food. Is this a sinister secret being kept from us? Or is it a fundamental misunderstanding of basic biology and scientific processes? Let's hunt down the truth. Part 1. A scientific breakthrough. Our story begins not in a shadowy corporation, but in a scientific lab in the Netherlands back in the early 1970s. Dr. Alex van der Ebb and his team at Leiden University were conducting groundbreaking research into human cell biology and viral transformation. In 1973, they took cells from the kidney of a legally aborted fetus. This wasn't an unusual practice at the time, and still today, various human tissues are used in research to understand human biology and disease. The HEK stands for Human Embryonic Kidney. The 293? That simply denotes the 293rd experiment conducted by Dr. Frank Graham, a postdoctoral fellow in van der Ebb's lab, who successfully transformed these cells with adenovirus DNA, making them immortal. And this is crucial. Immortalized means these cells don't die after a fixed number of divisions, like most normal cells. They can grow and divide indefinitely in a lab environment. The HEK 293 cells used today are not the original cells from 1973. They are descendants, generations upon generations removed, cultured, and propagated over 5 decades. No new fetal tissue has been used to create HEK 293 since that initial event. Part 2. A workhorse of biotech. So, why are these cells so important? Why have they become a cornerstone of modern biotechnology? HEK 293 cells are simply scientific workhorses. Their robustness, ease of growth, and ability to produce large quantities of proteins make them invaluable in medicine. They're used extensively in vaccine development and manufacturing, not as an ingredient, but as a tiny biological factory. For example, some COVID-19 vaccines, like Johnson & Johnson's and AstraZeneca's, used HEK 293 cells to grow the harmless adenovirus vector that delivers genetic instructions to your immune system. After the virus is grown, it's harvested and purified, removing the HEK 293 cells entirely. The final vaccine contains no cells, just the active ingredients and other standard vaccine components. Gene therapy and drug development. Researchers use them to produce therapeutic proteins, study gene function, and test new drugs. They help us understand how diseases work and how to fight them. To reiterate, when you get a vaccine that utilized HEK 293 in its production, you are not being injected with human cells. You are receiving a highly purified product. The cells are a tool in the manufacturing process. Like the molds used to make car parts, the mold is not part of the car itself. Part 3. In your food? Now, let us confront the most explosive claim, that HEK 293 is being secretly added to our food, especially fast food. This isn't just misinformation, it's a potent urban legend. The origin of this particular myth can largely be traced to a company called Cenomix. Cenomix was a biotech firm that aimed to develop flavor enhancers, compounds that could make things taste sweeter or saltier without adding more sugar or salt. Here is what they actually did. Cenomix scientists engineered HEK 293 cells to express human taste receptors. Essentially, they created tiny lab-grown taste buds. They would then expose potential new flavor compounds to these engineered cells. If a compound activated a sweet receptor, for example, the cells would react, giving scientists a rapid way to screen for novel flavor enhancers. Do you see the difference? The HEK 293 cells were a testing platform, a tool in the research and development phase. They were never, ever an ingredient in the final flavor compound, let alone the food itself. It's like using a specific type of microscope to identify a new spice. The microscope is not an ingredient in your meal. Despite the clear scientific explanation, various organizations and online communities seized on the connection, fueling claims of cannibalism and unethical practices in the food industry. They called for boycotts, pressuring major food companies that had partnered with Cenomix. In response, many companies clarified their positions or ended their associations, not because they were putting HEK 293 in food, but to appease public concern. The FDA, the regulating body for food and drugs in the United States, has been unequivocal. Food containing human cells or tissue would never be approved for consumption. The claims simply do not hold up to scientific scrutiny. Part 4. Why this myth persists. So why does this myth endure? It taps into potent fears of hidden ingredients, fears of corporate greed, and deep-seated ethical and religious objections to the use of embryonic cells, even for medical research. It's a classic example of how a grain of truth, the use of HEK 293 in a lab, can be distorted into a mountain of misinformation, amplified by social media and a lack of scientific literacy. HEK 293 is not a secret ingredient. It is a vital, immortalized cell line that has contributed immensely to our understanding of human health, the development of life-saving vaccines, and crucial medical advancements. Its origin comes from a specific historical event in science, but its current use is detached from that origin, propagating in labs worldwide, serving as a tool, never an ingredient. Understanding the science helps us separate fact from fiction, and when it comes to HEK 293, the truth is clear. It's a powerful research tool, not a hidden additive. Have you heard any rumors about food additives? So drop a comment in the box.